Welcome everybody. I'd like to start by acknowledging the traditional lands of the traditional owners of the lands whom we're meeting today. So for those of us at Monash University and Monash Medical Centre, that's the people of the Kulin Nations. And I'd like to acknowledge their traditional, the elders past, present and emerging. So you'll see the timetable of the session that we're going to be running this evening. I'm just going to start with the initial housekeeping to show you that this is the speakers that we have today, and that we'll have a few minutes at the end for questions. You can pop your questions in the chat during the presentations, and we'll either answer them at the end, or some of the speakers might answer them as we're going through the presentations. So, and just a reminder to place yourselves on mute. So I will pass over now to Professor Stuart Hooper, who is the head of the Ritchie Centre. Thanks, Stuart.
Thanks, Megan. Do you want to start our slides? Look, it's good evening, everybody. It's my pleasure to be here. I'm the head of the Ritchie Centre, and it's my job just to give you a little bit of an overview of the centre. The centre is actually focused on improving women's and newborns' health, and we have 6 themes which are focusing on women's health before, during and after pregnancy, fetal and neonatal health in 3 regions, perinatal infection and inflammation, cell therapy and regenerative medicine, and in newborn imaging. Next slide, please, Megan. We offer a diverse range of projects and expertise. We do lab work, we do big data, and we also do clinical studies. Next, please, Megan. And we offer projects in a variety of different areas. You can study pregnant women, you can do cell culture studies, mice studies, studies in babies, or indeed, we do a lot of studies in lambs. Next, please, Megan. We work at all stages along the continuum between women's health, pregnancy and baby health. We do research into women's health before pregnancy, so treating endometriosis, during pregnancy and after birth. We're looking at stillbirth during pregnancy, stillbirth rates, treating preeclampsia, effect of maternal obesity, and a variety of other pregnancy disorders. We use stem cells to try and prevent or cure disease, stroke, lung disease, etc. Next, please, Megan. We do a lot of work in pre-birth and birth transition and preterm birth, focusing on how to aerate the lung and when should you clamp the umbilical cord, a variety of different projects. And you'll hear about these more from the supervisors in the next couple of talks. Speaking of which, so I'll move on to the first one. Next, please, Megan. I'm here to talk about the respiratory theme. And in that, next, please, Megan, we do lots and lots of projects, and you'll see that we do a lot of imaging in this research. And I've put up here, in the first instance, we look at preterm infants, how to aerate their lungs, and what you're looking at is a preterm rabbit gradually aerating its lungs with each inflation. And we look at how to stimulate them to breathe. Next slide, please, Megs. We look at, well, how do we transition and improve the transition to newborn life in infants with a congenital diaphragmatic hernia. These are 2 CT scans or 3-dimensional scans of lungs from rabbit pups with a congenital diaphragmatic hernia. And you can see that one with the CDH has a much smaller lung. And as a result of that, they don't readily transition to newborn life very well. Next slide, please, Megs. We look at the effect of caesarean section delivery on newborn health, and in particular how they aerate their lungs. There's lots of evidence that if you are delivered by caesarean section at term without labour, then you're at much higher risk, or 2 to 4-fold increase in risk of developing respiratory distress in the newborn. We're trying to work out why. And these are 2 images. One's a control and the other one is delivered by caesarean section. And you can see the lung is not as well aerated. Next slide, please. We're using very sophisticated 3D imaging techniques to track liquid as it clears the lungs at birth and try and work out the mechanisms by which that occurs. So that's more discovery type of research rather than being directly applicable to the clinic. Last one, please, Megs. And we look at the role of surfactant. How well does surfactant aerate the lung? Many infants are born, and particularly preterm infants are born, and they only aerate part of the lung. So it's a big challenge for them to aerate the entire lung. Just imagine that you're a clinician and you're trying to ventilate these little infants, and all of the air is just going into the aerated region, not into the unaerated region that you can see there. We're looking at how surfactant can improve those. So these are all the types of projects that we do in the respiratory theme. A lot of it's all about improving that transition from a fetus to a newborn, where the biggest challenge they face is how do they aerate their lungs. Okay, that's it from me.
Thanks, Stuart. Our next presenter is going to be Professor Susie Miller, who's going to tell you about the neurodevelopment and neuroprotection research theme. Thanks, Susie.
Thanks so much, Megan. Hi, everyone. I'm Susie. I'm a Deputy Director of the Ritchie Centre, and I head up the neurodevelopment and neuroprotection research theme within the centre. So essentially centred around brain development and what happens to the brain when things go wrong. We know that in Australia, about 10% of pregnancies are impacted by complications that can alter brain development. Forward, please, Megs. And essentially, these are categorised into 4 main compromised situations. These are fetal growth restriction, intrauterine inflammation or infection, preterm birth, and perinatal asphyxia. Now, at the Ritchie Centre, we study all of these complications. We have animal models and in vitro models, and we're interested in the effects of all of these on the developing brain. So then our team works to reveal the cellular mechanisms that mediate fetal and neonatal brain injury so that we can test and implement targeted therapies to prevent long-term neurodevelopmental deficits, including the motor deficit cerebral palsy. Forward and forward, thanks. I'm just going to give you a really small snapshot of some of the projects that we've got going on. I encourage you to speak to any of the people or myself that I'm going to put up on the screen next, but I just want to give you a little snapshot of some of the brain projects, brain-related projects within the Ritchie Centre. The first is a project by Dr. Emily Cam, and she's really interested in the interaction between the placenta and the developing brain. And one of her projects is centred on whether or not maternal obesity alters placental function, and in turn, what impact does this have on neurodevelopment? The second project that I'd like to encourage you to have a think about is how might stem cells be used to improve brain development? And in particular, this is Courtney McDonald, and Courtney leads a really large team looking at umbilical cord blood cells in particular, and NSCs that you're going to hear about from Maddie, and their potential role in neuroprotection in the neonatal period. You're also going to hear from Beth Allison next. Beth is really interested in the cardiovascular system, but as part of our group, we're trying to understand what the interactions are between altered cardiovascular function and altered brain development. So we have some really neat projects, in particular looking at fetal growth restriction and adaptive responses of the cardiovascular system during fetal development and their impact on the brain. This is me, and we've got some projects that you might be interested in. In particular, we're looking at the behavioural deficits associated with fetal growth restriction and neuroprotective therapies. These projects are predominantly being undertaken in newborn lambs, and we do a series of maze tasks in the newborn lambs to assess whether or not there are functional deficits associated with fetal growth restriction and whether or not we can match brain structure to function. And finally, down the bottom is Tamara Yorno. Dr. Yorno has some really interesting studies that are centred around what happens when things go wrong immediately around the time of birth. In particular, she's interested in altered brain activity that can lead to neonatal seizures. The current treatment for neonatal seizures is phenobarbital. Phenobarbital was introduced in 1912 and has been in use ever since. Now, it's not a particularly effective anti-seizure medication, and it's not particularly nice for the developing brain. So TAM's really interested in looking at new treatments that might be better for the developing brain and better at treating seizures. There's just a snapshot of the projects that we've got going on in the brain world at the Ritchie Centre. I'd like to encourage you to have a look at the student handbook and look at all of the different supervisors and all of the different projects that might take your interest. Thank you.
Thanks, Susie. Our next presenter is going to be Beth Allison. Beth, Susie just mentioned Beth, and Beth's going to talk to you about projects in the perinatal cardiovascular field and physiological field. Thanks, Beth.
Thanks, Meg. So yeah, as Susie just really nicely introduced the work that I'm really interested in, and I'd like to echo Susie's thoughts about getting in contact with us or anyone who you read projects that you might be interested in. We can always point you in the direction of new people to talk to, and it's really nice and a good idea to just get a really wide understanding of what the projects that are on offer, and have a good chat to the honours students as well. So in terms of the work that I'm really interested in, I'm interested in cardiovascular physiology, and I'm interested in this across this really wide spectrum. So looking at both the period whilst they're in utero, as well as the immediate neonatal period, and then into the first weeks and years of life. So we know that the developing cardiovascular system obviously it's important to get that right during pregnancy. And so I'm interested in how the actual uterine environment can impact the developing cardiovascular system. And there was some of the work that Susie was just talking about, where we look at fetal growth restriction and how this changes the developing cardiovascular system. Which brings me to my second point, which is during the neonatal period. Sorry, Meg, can you just go back? Which then we think about how the changes in the cardiovascular system, which we know occur during birth, how that's controlled and how the body is able to regulate that change at birth, which is critical for that fetus to become a neonate. I'm then really interested in how being born with an altered cardiovascular system, so for example, formed fetal growth restriction, can impact infants who might end up in the neonatal intensive care unit or the NICU. So how does it impact on the therapies that are being delivered or the type of ventilation strategies that are being used, for example? And then in the long term, I'm looking at whether there can be treatments which can reduce the burden of cardiovascular disease. And in this next slide, I'm just going to show you some really neat work which my most current PhD student has just popped on my desk. And don't worry too much about these graphs, but it's just showing you the evolution of cardiovascular disease in fetuses that are either growth restricted in yellow or control in blue. And we can see that we're already seeing some really interesting differences in this work. And so there's lots of new projects that are coming out of this, which we're going to be investigating. So if any of this piques your interest, please drop me an email.
Thanks very much, Beth. Our next presenter is Dr. Caitlin Philby, and she'll be speaking to you on behalf of the Women's Health Research Team in the Ritchie Centre. If you can unmute yourself. Thanks, Caitlin.
Thanks, Megan. Yeah, so I'm Caitlin Philby. I'm a postdoctoral researcher working in the lab of Professor Caroline Gargett, and I'm representing quite a large number of scientists tonight in the women's health theme. Megan, could you just move to the next slide, please? So we chopped off the title there, but the women's health theme is really underpinned by the discovery by Professor Caroline Gargett, our leader, of the presence of adult stem cells within the endometrium. And Caroline has since gone on to characterise each of these and their progeny. And it's these stem cells that we think regenerate the inner lining of the uterus with each menstrual cycle. And so they obviously play an important role in endometrial health and the ability to support pregnancy. But we also believe that they play a role in certain gynecological diseases, such as endometriosis, adenomyosis, and potentially fibroids as well. And all of these conditions suffer from diagnostic delay. They're often silent conditions that are hidden for many years, that women suffer for a long time before they receive treatment. And the current treatments are also less than optimal. So the endometriosis and adenomyosis arm of the program is really looking at how we can diagnose the disease earlier and also using various models, including in vitro models such as organoids and in vivo models such as the mouse model of endometriosis, which is led by Dr. Fiona Cousins, in order to understand how the diseases occur in the first place so that we can reduce the time to diagnosis and offer alternative therapies to these women. There's a wide range of techniques that we use in that program, including in vitro culture such as organoids, flow cytometry, FACS sorting cells, immunofluorescence, characterization of exosomes, which are extracellular vesicles that are produced by stem cells and other stem cells. Whoops. We'll just go back. And in the other arm of our work, which is the study of pelvic organ prolapse, we're actually trying to harness the endometrial mesenchymal stem cells as a cellular therapy for this disorder, which again is a silent condition that women have suffered, and many people think that this is a condition that affects much older women, but it can affect women as young as 30 and 40 after childbirth. And many of you would have heard about the synthetic meshes that have been pulled from clinical use. And so our research led up by Dr. Shayanti Mukherjee is really looking at, can we combine the use of stem cells and biodegradable meshes that work more in harmony with the woman's body, and perhaps even can we prevent some of the damage from occurring in the first place. And so there's a number of techniques that are employed in this area, including bioengineering, cell culture, small and large animal models, immunomodulatory techniques, and both sides of the research are really well funded both by the NHMRC and international funding, and have really strong clinical collaborators as well as other collaborators such as clinicians and epidemiologists and geneticists. So if any of this sparks your interest, please do get in contact with Professor Caroline Gargett or myself or Shayanti Mukherjee, who's leading up the pelvic organ prolapse, or any of the other postdocs such as Dr. Fiona Cousins, Dr. Shanti Gurung, who's working on exosomes with Dr. Tamas Tatmaya. Thank you.
Thanks, Caitlin. So our next two presenters are now speaking on behalf of different student bodies within the Ritchie Centre. So Maddie Smith is a current PhD candidate who's come through the Bachelor of Biomedical Science Honours pathway, and she's going to speak to you, give you a snapshot of what it's like to be an honours and PhD student within the Ritchie Centre. Thanks, Maddie.
Awesome. Thanks, Megan. I'm Maddie and I'm a third year PhD student at the Ritchie Centre, and I'm a part of the Miller Allison Lab Group, who we've just heard from Susie and Beth tonight. Next slide, please, Megan. So I just wanted to start with a bit of an overview of how and why I chose the Ritchie Centre. So as Megan said, I did biomed as my undergrad, and in my third year I did the course BME Fetal and Neonatal Development, and that is a subject that's based at the Ritchie Centre, and I was really blown away by that subject. It really sparked my interest in research, especially the large animal facility that we have that houses the sheep, and other aspects including the way that we're really tied to the clinical world, and doctors are our colleagues. So it's really motivating, the environment that we're in, and it's a really exciting environment to be in as a student. Next slide, next little thing, yep. So I wanted to provide potential students with some advice, and that's to find a project that excites you. And that might sound a bit cliched, but you're dedicating a year or more to your life to that project. So I think it's really important to do some background reading and think about the field before you meet with your potential supervisor and have any questions prepared. Next one. I found the Ritchie Centre to be a really supportive environment. Everyone sits together in a large open plan office, and it's really easy to make friends. And I've made friends for life at the Ritchie Centre, which has been amazing. Next one. And then the next one. So at the Ritchie Centre, we're a part of the Hudson Institute Student Society, which has a lot of social opportunities as a part of that. So there's trivia nights, movie nights, there's netball, the 3-minute thesis competition, dumpling nights, the research week symposium. So there's lots of opportunities to present your work. And there's a student retreat, which was a highlight for me, where everyone got to go to Phillip Island for the night. You got to know people in a more casual setting than at work, and you got to present your work. It was a really great environment to be in. And obviously, hopefully, if COVID goes away, we'll be able to do these things again sometime soon. I've absolutely loved my time at the Ritchie Centre, and I encourage everybody to email potential supervisors and look into everything a bit more. Thank you.
Thanks so much, Maddie. So as Stuart suggested, we're a really translational research centre at the Ritchie Centre. So we have a large proportion of our students who come through from a Bachelor of Science, Bachelor of Biomedical Science degrees, but we also have medical students who come through as BMed Honours students, and a lot of practicing clinicians who come to do their PhD with us. And Shiraz Badruddin is going to talk, is one of those candidates. So he's already a clinician. It's about his experience of doing his PhD as part of his research fellowship in neonatology. Thanks, Shiraz.
Thanks, Megan. So yeah, as Megan said, I'm a clinical fellow in neonatology, just coming towards the end of my PhD that I did with the perinatal transition group. So I had the opportunity to do both clinical trial in babies, but also some LAM work on perinatal asphyxia. And I also had an affiliation with the Royal Women's Hospital, so it was nice to have both clinical and science-based supervisors. So I'm a UK-based trainee, but I came to Melbourne to do this PhD really because my supervisors are recognized around the world as leaders in this area of research. And it was a way for me to get on the pathway of becoming a clinician scientist, which was sort of my career aspiration, and sort of establish those worldwide networks in this area of research. Monash and the Ritchie Centre are a great place to do a PhD in maternal fetal newborn science and research, because it really seems to be an area that's growing at the Monash site and the affiliated institutions. It was really important for me to have supervisors that had previously supervised clinicians, because they really knew how to strategically prioritize my career progression, both from a scientific point of view as well as from a clinical point of view. And also to have those supervisors that would allow me to try and carve out some research questions that I found really interesting within the broader scope of work that they were doing in their groups. The research training opportunities are there. They extend beyond the Ritchie Centre, and they encourage anyone who's thinking about doing a PhD to really look into what you'd walk out with at the end of your PhD in terms of transferable skills, not just the topic expertise that you might gain. And that can range anywhere from, learning to code, to advanced statistical methodology, to learning how to launch a startup. There's opportunities to pick up those skills during the time, and it's really worth investing some thought into that and talking to lots of people about how you might do that. Finally, an attraction was to be able to do a PhD with published works rather than with a thesis, because I felt that would allow me to have some papers by the time I finish my PhD and help me with my academic progression. So I'd highly recommend it. Happy to talk to anyone that has any questions about the process.
Thanks, Shiraz. So now I'm just going to take a few minutes to talk about how you might apply for honours and PhD. So my name's Associate Professor Megan Wallace. I've probably forgot to introduce myself at the start. I'm a member of the Ritchie Centre and also the Director for Medical Student Research for the whole Faculty of Medicine, and also the Chief Examiner for BME 3082, which was the undergraduate unit that Maddie Smith mentioned. So the Ritchie Centre this year, we currently have 42 PhD students, a real mixture of people coming from science background and clinical background, as we just mentioned. We've currently got 30 honours students across a range: Bachelor of Science, Bachelor of Biomedical Science, and Bachelor of Medical Science honours, and 1 master's student. What will you get from research training at the Hudson Institute? So we've already touched on some of these things during the prior presentations. First of all, a qualification that demonstrates that you're really highly skilled in biomedical research, advanced critical thinking, really excellent writing and speaking skills. I think our students are really outstanding in this aspect. We give them fantastic training. They stand out when they present at conferences. You get training in state-of-the-art research platforms and colleagues who are really dedicated to improving human health and well-being. If you're somebody considering applying for honours, then we suggest that you find a supervisor with a project that interests you. I will say, and I think probably everybody on the panel here today will agree, that the supervisor is actually probably much more important than the specific project. Find a broad area that you're interested in and then speak to the supervisors. We've got loads of fantastic supervisors in the Ritchie Centre. Send them an email, introduce yourself, check if the project is still available. I would do that initially via email. Supervisor Connect is a great way to find projects. Set up a Zoom time to meet with the supervisor and their team, and if we're not in lockdown, come in for a tour of the lab space, and if you're excited, then sign up. So students who are doing applying for Bachelor of Biomedical Science honours can speak to Mark Hedger. Students from a Bachelor of Science honours, speak to the honours coordinator from the department that you're applying through. A lot of our students come through anatomy, or if you're a medical student, then speak to me. If you've already done an honours or you've graduated with another degree and want to come back, like Shiraz, and want to come back and do a PhD, similarly, find a supervisor in a project, make sure that the supervisor and the research team are a good fit for you. Apply for the PhD, you've got a number of extra steps that you need to go through, particularly if you're an international student. You need to complete English proficiency, research qualifications, etc. There's several scholarship rounds. The main one is the 31st of October, and Caroline Gargett is the contact for Department of Obstetrics and Gynecology, and Rosemary Horn for Department of Paediatrics. I can't remember if Stuart mentioned that the Ritchie Centre is the research arm of these two departments in the university. This link will take you to a list of currently available honours and PhD projects in the Ritchie Centre. Okay, so we'll open the last minutes up now for questions. If you have got a question, click on the participants button at the bottom of your screen, click on the participants button and raise your hand, or use the chat function at the bottom of the presentation to type in a question. I will stop sharing, and please feel free to turn on your cameras, turn yourself off mute and ask a question. So Georgia, I can see that you've got your hand.
Hi. So I've actually found a supervisor in a project already, and I just wanted to know, how do you determine what stream you're supposed to do it through? So like, I'm doing a Bachelor of Science, and I think I can either do physiology or anatomy. How do I know which one's best to select? Because I started my application, but I'm not too sure what to submit. And is there anything I have to do, like paperwork I have to do through the Ritchie Centre? Because on the Monash website, it directs you to email you guys first. So yeah, sorry, that was a lot at once.
No, that's okay.
Megan, I can answer that if you like.
Go for it.
I think, Georgia, I think you'll have to go through Department of Anatomy and Developmental Biology. There is a requirement, if you go through physiology, that we actually have a physiology supervisor there. And so I think you'll probably find it'll be easier to go through the Department of Anatomy and Developmental Biology.
Okay, thank you.
And you just have to get agreement with your supervisor about your project and sign off from that. Okay.
Yeah. So yes, I've got that approval, and then I can just apply through e-admissions.
Yeah.
Cool. Thank you.
Okay. So Chrissy, Chrissy, you can turn yourself off mute if you'd like, otherwise I can read out the question.
Hi, yeah. So I just wanted to, really random question. I was just thinking about it while you were talking. So with PhD, it's not even relevant to me, but I just wanted to know, is it a prerequisite to do honours before you do PhD, or are they could hypothetically someone jump from undergrad into PhD? Because I feel like that's not a thing.
Yeah, that's right. So if you're science or biomedical science or straight out of or doing your medical degree, then honours is a prerequisite for a PhD. Or you can, if you have several publications, so sometimes people might work as a research assistant for a few years, or if they're a clinician, they might publish papers during while they're a practicing clinician, and then you can use those papers to gain what's called H1 equivalence, so to get equivalence of a first class honours degree.
Ah, that makes sense. Thank you so much for that.
No problem. Okay, Gabby. Gabby is asking, what's the main difference between the Ritchie Centre and the Centre for Reproductive Health? And
do you want me to answer that one again, Meg?
Sure.
Yeah, look, the Ritchie Centre is the only centre that does fetal and neonatal health. There is a little bit of women's health that is also done in the Centre for Reproductive Health, but they do mostly, well, not mostly, but they do a lot of male reproductive health as well. So they don't do any fetal and neonatal research, and they do a little bit of women's health, but nowhere near as much as what we do.
Great, thank you.
And Jasmine Yap is asking about the open tour form. I suspect that the open tour form might be a hangover from last year, or else they were hoping that the lockdown would have lifted by now. So if you would like to come and visit the Ritchie Centre, just send us an email. If you go to the Ritchie Centre booklet, you can email anybody in the booklet. You can email myself, megan.wallace@monash.edu, and we will arrange a time for you to come and visit us once lockdown is finished. So to get into the Ritchie Centre, you actually have to walk through Monash Medical Centre, and we have to go through a whole series of processes before we can bring people through that at the moment. So, and Emily's asking, is honours a prerequisite for masters by research? That will depend on the master's degree, I think, Emily. I think you'll need to look up this individual requirements for the master's degrees. Different master's degrees have different entry requirements. Is it possible to do 3990? Gabby, yes. We definitely do 3990. In fact, Susie Miller mentioned Emily Cam in one of her presentations, and Emily is actually the 3990 coordinator for the Monash Health Translation Precinct. So if you're interested in doing 3990, then Emily Cam is the person to speak to. And yes, we often have 3990 students on site. Any more questions? You can just pop your hand up. We were probably lucky to be the last session of the day, so we've managed to go on probably a wee smidgen longer than we should have. But while you're thinking about any extra questions, I'm just going to quickly pop up one more slide about the sessions that are coming up over the next, coming up tomorrow. But in the meantime, raise your hand or I will probably just turn yourself off mute and ask the question. So here we are tonight at the Ritchie Centre, the last session of the day, but I'd also like to draw your attention to the sessions that are on tomorrow evening. So as I said, the Ritchie Centre is the research arm of the Monash, so it's the part of the Hudson Institute, but also the research arm of the Department of Obstetrics and Gynecology and the Department of Paediatrics within the university. So we've got two sessions tomorrow, one at 5 p.m. and one at 5:30 p.m, talking about some of the other types of research that we do within the Department of Obstetrics and Gynecology and within the Department of Paediatrics. So please feel we'd love to see you again tomorrow night if you'd like to come and join those sessions. Okay, I don't think there's any more questions. So otherwise, if not, I'd like to thank all of our presenters this evening for keeping to time, doing a really great job actually of keeping to time. I'd like to thank all of the students who've joined us today for coming along and hearing about what the sort of research that we do, and also for asking those questions. Thank you.
Thank you.
Thanks, Megan. Thanks, Shiraz. Thanks, Mads.
Thanks, everyone.
All right. Great job. Thanks, Meg. See ya.
Okay, thanks, Suze.
Thanks. Bye.
Thanks, Meg. See ya.
See
I'll
ya.
see you tomorrow night.
Yep.